and the relative properties of various body parts depends on the temperature. For homotherms and for poikilotherms, there are two different sets. For homotherms, in cold regions, some birds and mammals attain a greater body size than in warm region, and larger species are common in colder areas. These are for homotherms. And for poikilotherms means cold-blooded animals. This changes in reverse order. There are smaller in colder region. Poikilotherms are smaller in colder region. Body size has played a significant role in adaptation to low temperature. Larger wood rats can survive in colder climate because their surface to air ratio and greater insulation permits them to conserve metabolic heat. Small sized animals are favored in desert. Allen's rule. It is different from Alley's rule. Alley's rule says that there is a decline in the population if their population density decreases, and some small population has risk of extinction, that is metapopulation type of has the risk of extinction. Allen's rule states that extremities of organisms such as legs, tail, and ears of mammals are shorter in colder climate. Some mice reared at 31 degrees Celsius to 33.5 degrees Celsius have longer tail than those of the same strain reared at 15.5 to 20 degrees Celsius. Means mice reared at the higher temperature have longer tails than in colder temperature. RENSCH'S rule. In colder region, the races of the bird have relatively narrow and more acuminate, means tapered to a slender point, acuminate wings, whereas those in warmer climate tends to be broader wings. Next, Jordan rule. Temperature also influences the morphology of certain fishes. Some fishes have a relation to their number of the vertebrae with temperature. Cod fish, which hatches off at a temperature between 4 degrees Celsius and 8 degrees Celsius, that is near Newfoundland. It has 58 vertebrae, while that hatching at the temperature between 10 degrees Celsius and 11 degrees Celsius, that is east of Nantucket, has 54 vertebrae. Means colder temperature cod fish has more vertebrae, and slightly hotter temperature cod fishes have lesser number of vertebrae. This is Jordan rule. It is the table that is related with the chromatin and their packing ratio. Different states of chromatin and their packing ratio. Naked double helical DNA. It has packing ratio equal to 1.0. 10 nanometer elementary fibril of nucleosome, it has packing ratio 7 to 8. 25 to 30 nanometer chromatin fiber of superhelical nucleosome, it has packing ratio of 40 to 60. Condensed metaphase chromosome of the loop, it has 8000. So maximum packing ratio is metaphase chromosome. There are some enzyme markers of different membranes. It is very important question. Plasma membrane has three particular enzymes: 5' nucleotidase, adenylyl cyclase, and sodium potassium ATPase. These are important for plasma membrane. Endoplasmic reticulum has a special enzyme marker of glucose 6 phosphatase. Golgi apparatus have different portion. In plants, it is called as Golgi networks. Golgi apparatus cis has GLC-NAC transferase 1. Medial Golgi monosidase 2nd. Trans part of the Golgi apparatus galactosyl transferase. TGN sialyl transferase. This table show cloning capacity of common cloning vectors, and there has been one question in perhaps 2014 or 15 from this table. Vector and DNA insert size. Each artificial chromosome, it can insert 500 to 300 kilobases. Bacteria artificial chromosome P1 52 to 50 kilobases. Cosmids 35 to 50 kilobases. Lambda charon 4A 10 to 20 kilobases. Plasmid PBR 322 0.01 to 10 kilobases. So if we see, this is the descending orders from yeast artificial chromosomes to plasmid PBR 322. Next table is Cope rules, and Cope rule is related with the evolution. Organisms have a tendency towards increase in size during their evolution. For example, evolution of horse, camel, and all other herbivorous mammals, they exhibit increase in the size, and some of them even attain gigantism. So this is not the universal rule. Cope rules also have some exceptions. A number of the mammalian lineages do not show the tendency towards increases. For example, insectivores and some insectivorous bats. Dollo's law of irreversibility. Dollo's law of irreversibility states that once such a complex state has been lost, it cannot be regained. Means complex state once lost cannot be regained. Oviparity is an example for such a complex state and has been lost on several independent occasions throughout the animal evolution. This is the table relating with the enzymes, their reaction and uses. Sorry. Alkaline phosphatase enzyme, it is used to prevent self-ligation by the removal of 5' phosphate group. Reaction, it catalyzes 5' and dephosphorylation of RNA and DNA. DNA polymerase 1, double-stranded DNA from single-stranded DNA. It catalyzes this reaction. Generation of blunt ends from sticky end and nick translation. Lambda exonucleases. Removal of nucleotide from 5' end of the DNA, and it is utilized in DNA sequencing. S1 nucleases, single-stranded DNA degradation, removal of hairpin in synthesis of complementary DNA. This table shows some genes, chromosome on which they exist, and if there is any disease associated with it. DNA segment G8. It is on chromosome number 4 and it may cause Huntington chorea. Insulin, that is, insulin genes are located on 11th chromosome. Alpha globin on 16th chromosome, and they may cause defects in alpha globin genes may cause alpha thalassemia. Prolactin on 6th chromosome. Beta globin 11th chromosome, and if there is any defect on beta globin genes, beta thalassemia and sickle cell anemia. Hypoxanthine guanylyl phosphoribosyltransferase transferase. It is located on X chromosome, and defects in any of this gene, Lesch-Nyhan syndrome. Phenyl hydroxylase. This gene is located on 12th chromosome, and defect cause phenylketonuria. Adenosine deaminase located on 20th chromosome, adenosine deaminase deficiency. Growth hormone genes are located on 17th chromosome. This table show localization, and there is a small small RNA. In nucleoplasm, there are 3 small RNAs that is U2, U4 and U5. In nucleoplasm and heterogeneous RNA, U1. In nucleolus, U3. Perichromatin granules, U6. Nucleus and cytoplasm, that is 7S.